Hello everyone, high biology DNA structure today. So the structure of DNA stands for, it's a ribonucleic acid, a deoxyribonucleic acid, okay? Equals deoxyribonucleic acid. So it is a complex molecule. Okay, in all living cells, okay, stores genetic information in its sequence of bases, okay, which these determine the genotype of an organism, okay, and the structure of proteins, okay. So DNA is made of nucleotides, okay. The structure of a nucleotide is what's the structure of a nucleotide? It's a phosphate, okay, attached to a and then a base, okay. So that's a phosphate. Okay, this is the sugar. Okay, and this can be A, T, C, or G. Okay, any of four bases. So you've got the deoxyribose sugar is a okay, so that's 1, 2, 3, 4, 5, okay. The 3 carbon is normally called the 3 prime carbon. And the 5 carbon equals the 5 prime carbon. In the DNA structure, you hear about 3 and 5 prime. That's where that comes from. The 3 and the 3rd and the 5th carbon of the sugar, the deoxyribose sugar. Adenine, okay, that equals cytosine. G equals guanine equals thymine. Okay, the structure of these, to get sort of this is diagrammatic, but these are complementary, okay. So adenine is complementary to that, fits into that, okay. And So that fits with that one. Okay. And that one fits with that one, okay? So that double bonds to T and that triple bonds to G, okay? So I drew out the, when I drew this, which is a phosphate surrounded by carbon 3 that's the third carbon. At least pull that in. So this is simplistic. Okay, but it's the overall shape, but these all are more structured than more structure. This is a simplification. So these are joined up. You can show them like this. Okay. Okay, that illustrates how they link together. I'll keep it consistent with doing that, okay? So this part forms this part here. Try and draw a bar. I made a mess of that, didn't I? Okay, that is the sugar phosphate backbone. Okay, this part up to, okay, these are the bases. This is the sugar phosphate backbone, okay, the phosphates and the sugars. So yeah, so the actual structure of DNA can be drawn as that should be the phosphate is better. Okay. Okay, so you got okay. Now the way you draw the other side is to turn it over. Incredibly, because it's just the reverse. This side goes 5 prime, okay, up here, and that is 3 prime, okay? That's the 3 prime carbon here. Okay, maybe I should draw that. That refers to that carbon, okay? So to do another complementary strand, you just turn it around. It's as simple as that. And you do this. If I can get this right. guess it takes a bit of practice, but I've been doing it longer than you, so I used to be able to do this pretty easily. Let's see if I've still got the knack. So you go 1, 2, 3, 4, 5. What am I doing now? It's not so easy, is it? It's not so bad. He's got to repeat it. Okay? And you gotta... Okay. So this is, again, that's the 5' carbon, and that's the 3' carbon, okay? So I could turn it around, it's exactly the same. Told you, it's the same. So yeah, this is why that's 3' to 5'. So if that's the end of the molecule, this is 5', and that's the end, that's 3', okay? That's where it stops. It can't stop. If it stops there, it's also 3'. Okay, it can't stop in the 5', otherwise you'd have a phosphate hanging like that. Okay, see? So if it ends in on the sugar, it must end in 3'. So that's why one direction is going 5' to 3', and the other one direction goes 3' to 5'. Follow that? So yeah, it ends if it ended on the phosphate, it can't do that. It has to end on because the whole nucleotide is present. It has to end on the sugar. That's why it ends in the 3'. Why the other end ends in the 5'. It cannot end on a phosphate. Okay, otherwise it's just the chemistry is wrong. The physics and chemistry doesn't allow that. So because the whole nucleotide is present, the last carbon, that going upwards, is 5', and the bottom one's 3', okay? I just want to make that clear as I'm thinking about it. And these are double bonds, and these are triple bonds, okay? And these are hydrogen bonds. And all the rest of them are covalent bonds. Okay? So that's the structure of DNA. And these are the base pairs. So we've got, yeah, and because they're 5' to 3', so they're 5' to 3' and 3' to 5' are called anti-parallel strands, okay? Okay. And the whole thing gets, so this because of the way the atoms are all joined together, it's not linear like this. It's not linear. I mentioned in some chemistry videos what linear is. This is not linear. It gets twisted into a double helix, okay? So it forms a double helix, which you can draw kind of like that. Okay? And you can represent sort of the base pairs like that. Okay? Quite a lot of my students struggle to draw these shapes and struggle to draw this. I haven't been doing this a long time. This wasn't through one attempt, it was multiple practices. You get used to it. do one side and then turn over and do the other side, and amazingly you draw the structure of DNA. That's how you do it. So that twists around for double helix. Okay, so these are the base pairs. And the sugar phosphate. Backbone. Okay. I don't think you have to know why it's called deoxyribose in this course. It doesn't actually say it, so I'm not going to go into it. It's a shame, but you don't have to know it. And then the DNA replicates. So maybe I'll do net replication next video. Otherwise we've gone too long. So yeah, hope you find that very helpful. I could draw that as 5' there. 1, 2, 3, 4, 5, 6, 7. 3', 3', 1. I could go to that one and checking it though, in case I made a mistake. That is indeed 5', okay? So that's how you draw it. So as I said, it stops at the carbon, 5', 3'. It has to end on a nucleotide, okay? It cannot end on a phosphate. If it ended on a phosphate, then they would both be 3' or something. But because it ends on a nucleotide, the last carbon at the end is either 5' or 3'. That's why one strand is 5' to 3', and the other one is 3' to 5', and the anti-parallel, okay? And the chemistry, the geometry of all the chemistry and chemical bonds. So the geometry of all the various, you've got angles like this. I call that theta, okay? Theta, okay? All these geometries, theta, okay? And all the other bonds, angles, and even these, the geometry sort of theta, okay? All these different geometries, I'm calling them theta to illustrate their angles. All these different geometries on that angle there, they will cause the helix to twist in a double helix, just because of the geometry of all the chemistry bonds and the atoms and the pentose rings, the sugar rings, and the physics of it all. Electrons repel each other, charged atoms repel each other, phosphates are actually negatively charged. So it forms the double helix to form, okay? Like that. So if any of you are wondering, I've explained all that to you to help you understand why this happens, okay? Okay, good evening.